in true CGSI fashion, I will introduce myself. But first, I'll say, this talk, when I was thinking about what to chat about, it's like, there's a few different things I want to cover. What's the consistent theme? And at the end of the day, I realized the consistent theme is that these are all projects that we thought would be easy and straightforward, and that turned out to be hard. And I think there are some lessons in that. OK, so by way of introduction, within my research lab, the way I usually describe what we work on is that we're not unduly focused. But we usually think about machine learning, public data, and the transcriptome. If you pick 2 of 3, we're probably interested. In my role chairing our Department of Biomedical Informatics, my role is to support faculty who work to make serendipity routine to advance research and care. As informaticists, I think this is actually our job. I think we're in the serendipity business. I think it's a good business to be in. And then through our Colorado Center for Personalized Medicine, I work to bring genetics and paired decision support to the point of care. So there are hard things that we thought would be easy in many of these, but I'm going to focus on things from the research lab. And I think these challenges are the things that come up many times as we carry out experiments. I think sometimes when we go to talks like these and we see these projects, we only see kind of the end result of this amazing tool that works and seems like it's effortless. And I think under the hood, what you don't see is the enormous numbers of frustrated hours that actually go into producing that. I won't give you an enormous number of frustrated hours in this talk. I think I would get pulled off the stage by Elazar. But I'll give you an idea of the types of challenges that we encountered for a few different projects. So 2 of these that I'm going to mention today are sort of, the first one seems really simple. How are any 2 nodes connected in heterogeneous networks? We heard a little bit earlier about, I guess yesterday, about these types of kind of multipartite networks and knowledge graphs and their use in biology. And then the second one is, again, seems really simple. How does the tumor microenvironment contribute to high-grade serous ovarian cancer subtypes? So if we want to tackle the first one, this is a story that came out of a postdoc who joined the lab. So Daniel Himmelstein joined my lab, and he had, during his PhD, developed something called the HADIO knowledge graph. And there are now many sort of resources like this, but this is a knowledge graph that incorporates more than a few million different edges that connect a number of different biomedical entities. I think it was roughly 10-ish different biomedical entities, and there's about 27 different edge types that can connect them. And when Daniel joined the lab, he had just done a project focused on drug repurposing, where he used machine learning methods applied over this knowledge graph to try to predict potential repurposing opportunities. The simple question that I asked him to work on was, well, what if we didn't have a gold standard, right? Because if you need a gold standard, then it limits the number of sort of types of entities you can query, right? You have to be able to construct that gold standard. That seems like a pretty easy extension. I'm going to use this example throughout the talk. So through this portion, there's a drug, an anti-diabetic drug, metformin, that's inexpensive, widely used. Epidemiological studies showed that it also, well, there was a whole trail of evidence, but I guess by the time of this 2010 paper, suggested that it also had anti-cancer activity. And so you might say, look, I've got an EHR, this is just one example, but you might have an EHR system, and you might say, look, there's this compound. When people take this compound, this disease is seen less. Why might that be? So all we wanted was a really simple thing explainer. So conceptually, we wanted this world where you could put in some source node, you could put in some target node, and we can tell you why they're connected without having to use a gold standard. So we went to the pre-Occam framing of Occam's razor. So Ptolemy wrote, if you translate it, we consider it a good principle to explain the phenomena by the simplest hypothesis possible. So that's all we wanted, right? I just want, what's the simplest thing that could connect 2 nodes in this graph? And for this example, let's just make life a little bit easier. We're just going to say, look, let's simplify it to breast cancer. Why might metformin prevent or treat breast cancer? And we're talking about simple, so let's just keep the paths short, right? Let's just say any path less than length 2. That's probably simple, right? How many of those can there be? So there can be many. There's thousands of paths less than length 2, and they have lots of different types. It can be that metformin binds a gene that's associated with breast cancer. It can be that metformin upregulates a gene associated with breast cancer. It could be that metformin is expressed in a tissue that breast cancer localizes to. There's like thousands of paths through that. And then you could even ask, like, well, why is path length 2 actually that simple? And so this is the point where this project goes from like a one-line Cypher query to a research project. Cypher is essentially SQL for a certain type of graph database. It's a way to query graphs in a simple way. And so now we're going to have to we're going to have to refine our question. So instead of sort of what seemed like a really simple, straightforward question, we have to say, okay, well, now we want to explain the relationships between biomedical nodes by the simplest corrected hypothesis possible, right? Because we need to correct for the fact that some, certain types of paths are less informative than others. Like if knowing that, a gene is expressed in a tissue, well, there's thousands of genes expressed in that tissue, right? So that's not a terribly specific path. On the other hand, a gene being bound by a drug that interacts, a drug binding a gene that interacts with another gene that's tightly connected to a disease, that's potentially more informative. And even though it's a shorter relationship, it's kind of potentially a simpler explanation. And so this is kind of where we end up. And you can start to ask a bunch of questions, because you're like, well, how hard could this possibly be, right? Now we just need to know the degrees of the source and the target node, and maybe the degrees of the nodes in between. But it turns out the background degree distributions for each of these edges is also quite different. And it's not necessarily clear up front, which paradigm one lives in. So there are some where sort of certain nodes are relatively low degree, other nodes are high degree. And so we ended up doing this entire sort of sub-research project within the broader research project to understand if this degree distribution was like meaningful. Like, do you care that the degree is different? Or is it just, well, the degrees happen to be different, so take it into account and you're fine. And one of the key experiments that was done there, so this was work led by Michael Zietz, who was an undergraduate in the lab, was to say, well, let's say degree is not meaningful. So, yes, some things are higher degree, other things are lower degree, but that's fine, right? Those are true biological patterns. They don't have anything to do with kind of bias in data collection. If that's the case, if I take two different experiments and I look at the degree distributions of nodes across two different experiments, they should roughly be the same, right? A high degree node should still be a high degree node. A low degree node should still be a low degree node. This would indicate that maybe degree can tell you something is nonspecific, but it's actually still a meaningful concept biologically. So what you see on the left here are degree distributions for different types of networks. So this one's a protein-protein interaction network. And what we're comparing is degree distributions from systematic experiments, so things where someone wants to sort of genome-wide profile some process, or in this case, protein-protein interactions, or those derived from literature-curated resources. And so what you see here is there's actually a pretty big difference in the degree distribution. And we know that these networks are going to be biased towards this literature-derived space. So you might say, ooh, OK, so now my degree distribution, yes, they're different, but they're also sort of, the degree distribution is not itself meaningful of the biological process. It is the biological process plus the way that we actually perform and study biology, right? So there's two different parts of the data generating process. One that might be sort of true biology, but the other one that is just kind of how we do science. And so we do actually need to control for this, and that turns out to be a challenge. And we saw the same thing across the different types of biological networks that we looked at. As a control, we looked at the co-authorship network on bioRxiv, pre and post 2018. And so these should look the same, right? We don't expect there to be a change in how people co-authored papers before and after 2018, and we really don't see a difference there. And so this kind of, broad research question led us to this additional research question, which is like, is this, meaningful, or is there sort of some, bias in there? And so that's, I think a key problem that we then had to solve before coming back and solving the broader question. I removed a few slides since we're starting a little bit late. But I will say, just to get to the point where we could actually implement this, in addition to understanding the degree distributions, we also had to write 3 new software packages that would let us effectively compute in real time, do a lot of pre-computation that sort of supported that, and then also develop somewhat accurate permutation test that we could put a probability on. We could calculate a p-value under a null. And so this project that seemed really simple, like one-line cipher query as we envisioned it, to actually kind of rigorously answer this question and build a server that anyone could use to do this ended up being about a 6-year process. And so it's now available if you're interested in playing around with it. But I just thought that was a good example of how you can take this really simple question, it becomes a complex research question, which leads to another complex research question, and then only after you solve those series of questions can you go back and actually produce what you sought to do. So if you want to play around with this, you can play around with it. These papers are just coming out now. The first one's led by Michael Zietz, and the other is led by Daniel Himmelstein. OK, second kind of thing on the same theme, easy questions that turn out to be hard. This one will take us into single-cell biology. I know there's been a lot of kind of single-cell discussion, especially on the genomics tech side. In this case, we were just trying to ask a simple question, how does the tumor microenvironment contribute to ovarian cancer subtypes? OK, well, that's easy. If I want to study the tumor microenvironment, I can just do single-cell sequencing, right? How hard could that possibly be? Well, it turns out that I really want to understand these subtypes, and so these are a population-level phenomenon. You can do a power analysis and kind of figure out how big your sample size needs to be if you want to study these. We did this. This is about the world you can do on 1R01. This is about what you need for appropriate power if you believe there are 4 subtypes of this disease that have previously been reported. I'm a little skeptical of that, but if you're going to believe it, that's what you need for adequate power. So that's not going to work, at least with the type of funding I can apply for readily. So anyone have ideas for what you might do? Anything? Is there a way to take single-cell data and bulk data and maybe get something out of the bulk data that you couldn't get without the single-cell data? No? No one wants to factorize anything? OK. So there's algorithms that are called deconvolution algorithms, where you can try to use single-cell data as a reference to estimate proportions in bulk data. So now you can take your bulk RNA-seq data, take a small number of single-cell references, and try to understand the cell type distribution in the bulk data. So we designed a simple experiment to do this, where you take a number of single-cell profiles, and then you also do bulk profiling. You can sort of discover potential cell types and reference profiles, and then evaluate how well that works. And then once you have something that works well, you can go and sort of factorize or use other sorts of strategies on these large population scale resources to try to estimate cell type distributions across subtypes. So this is the simple experiment that you could do. OK, so we're done. This talk can end here, right? Because this is just now it's a technical problem, right? All we have to do is turn the crank. Unfortunately, no. So this is a project led by Ariel Hippen, who was a PhD student in the lab. With Ariel, the first thing we did as part of this was just to ask a really simple question. Just thinking about how this process actually works, the way we design the experiment kind of naively is that you have a tumor sample. You can do bulk RNA-seq on that tumor sample. This is usually done through rRNA depletion. And you can take that tumor sample, dissociate cells, and do poly-A single-cell RNA sequencing. That just happens to be what people usually do. And then you just do deconvolution. So this is the sort of the world. There's a small set of challenges here, which you can probably guess, given that I've highlighted a few things. One is, well, first, it's also helpful if you can pool your samples, because you'd like to know if there are batch effects in your single-cell sequencing. And so one question is, if you pool your samples, does that change anything? But the second part is, you've also got this dissociation step, which could change your cell type distribution potentially pretty radically. And you've got this mRNA enrichment difference, right? The single-cell methods generally are using poly-A selection. But when we bulk profile population-based resources, we're usually doing something called rRNA depletion. So the way we actually get the RNA that we want to assay is different, because there's a few different technical reasons. But essentially, if you're going to profile a population, you're usually going to profile FFPE tissue. And that breaks up the RNA, so you cannot then do poly-A selection, which requires that you just grab one end of it. And so we started to ask the question of, OK, how well does deconvolution actually work in this setting where we know there are these technical differences? So the first question is pooling. Does pooling matter? Again, in the interest of time, I hid quite a few slides. So I'll just give you the punchline for each of these so that you don't have to deal with the build up to it. So pooling actually works fine. If you do genetic demultiplexing, we didn't see a big effect from any of the sort of prep stuff that's done. So we have confidence that this isn't going to influence the deconvolution results much. If you do antibody-based pooling, I will say that that's not necessarily always the case. We have kind of one observation for one sample where the antibodies did not appear to bind with the same degree to certain cells in one of the tumors. So if your experiment supports genetic demultiplexing, you're good. If it's antibody-based demultiplexing, you may struggle a little bit. Dissociation. So this is a process where we're basically taking a tumor chunk and breaking it up. This actually, we observe quite a few differences here. So we see a lot of different transcripts that do appear to be cell type specific, some of which we can explain directly by our prep method so that we actually remove red blood cells, so we don't see red blood cell associated transcripts. But others that probably indicate or obscure information in the biology. So in this case, one of the key things that we see is that adipose genes, we see a lot more of them in tumor chunks than dissociated cells. And it's particularly that the tumor chunks that had large amounts of adipose genes now have very little sort of expression of those adipose genes after dissociation. This is known, right? Fat cells break when you dissociate. But for ovarian cancer, there's actually biological differences between tumor samples that are pulled from the omentum and other sites. And the omentum is fatty tissue. So you're basically eliminating a lot of the signal in your data that might be meaningful if you care to understand the biology of these subtypes when you're doing dissociation. So this type of challenge can then go back and influence your deconvolution results. And then there's also sort of results related to the RNA isolation method. One of the key things, which I ended up hiding this slide because of time, but one of the key things there is that mitochondrial reads, you end up with very different results depending on which bulk RNA depletion kit you use. So if you're using that as a strategy to estimate when your cells are dead, you're going to throw out a lot of cells that are fine in your single cell experiment. And then, so summaries. So this really simple project, we designed a pretty careful small experiment to understand what the impact of the actual data generating process was going to be. Demultiplexing, not a problem. However, a lot of deconvolution methods assume that your cell type reference profiles are complete. This is unlikely to be true for some tissue types, including this tissue type. And there's a lot of results in the paper where Ariel looks at them, sort of examines the methods and demonstrates that certain methods exhibit a very large loss in performance despite being very good when you have a complete reference. RNA isolation differences, there might be ways around this, but it's not generally something that's built into current methods. And it also appears to induce some changes that are going to be difficult to work on. And then your existing deconvolution methods, which are largely created and evaluated using data sets that are kind of not reflective of many complex tissue data sets, those results may not generalize to the setting that you care about. And so this helped us pick a method that appeared to be reasonably well performing. I think there are still, it pointed to a lot of ways to improve the method, but it also sort of helped us pick a method that seemed to be relatively well performing so that Ariel could publish that really simple paper that we originally envisioned and complete her thesis. So yeah, just the work that sort of Ariel put into this, the technical work that Ariel put into answering these questions is really in these first 3 papers. And then that quick and simple analysis that we originally envisioned that would get us to sort of the biology that we were studying, that's just this 4th paper. And so I think this is another story, right, that starts pretty easy, and then the analysis plan, doesn't survive first contact with the data. Okay, CGSI, like thinking a little bit tutorially. Why does this, how do you deal with a project where you think it's going to be easy and then it turns out to be hard? Because this is at least, hopefully more than half of you end up in this setting, right? Because that's where the interesting science happens. So here's what we do in our lab, right? So we write down our practices. So if you want to see everything we do in our lab, you can go to our onboarding repository. And that repository really describes a set of processes where we're trying to consciously break down large problems into small ones. Because it's really hard to solve a large problem. It can seem very insurmountable. But a small problem, if I know what I need to do each day, I can make progress on that small problem. So that's what we try to do here. So we have like an annual process, a triennial process, we have a weekly process, and we have a daily process. So our lab is very process-oriented. And then for any project, we end up trying to maintain good software practices. So we have code review. All the work is developed through pull requests. We recommend linting and automated testing. And the reason we do this is because if you've got a project that seems like it's going to be easy, and you're like, this is a quick one-off project, I'm going to work on it for the next couple weeks, and then I'll have a result. Well, you work on it for a couple weeks, and then you're like, oh man, this turned out to be pretty hard. I'm just going to put a couple more weeks into it. But I'm not going to do this rigorously. I'm just going to put a few more weeks into it because it's just a proof of concept thing. Well, now you're a month down the road. And then you keep doing that for the next month, and the next month, and the next month. And like 6 months in, you find yourself with a mess of code, little organization, and no clue how those things fit together or exactly what you've done. So I think because we don't know how difficult a project will be up front, it's important to essentially say, OK, go into every project assuming there's going to be complexity, and approach every project in the same way. And then I was once telling someone about the processes in our lab, and they told me about this paper. So if you want a lot more detail on this type of strategy for sort of surviving graduate school or your postdoc or your faculty position, there's this article called The Mundanity of Excellence that looks at stratification of Olympic swimmers. It's published in 1989. And it makes the argument that excellence is mundane, right? It's not about talent. So they observe that talent in swimming at that time follows a very peculiar geographic distribution. So if you look for talented swimmers, they're almost exclusively from Southern California at that point in time, right? So I don't think we believe that talent in swimming is localized to this area, but it does have really nice weather, and it generally has, in the 80s, was economically well off. And so you've got people with the net worth to put free time to get their kids into swimming, and nice weather in which to do it year round. So this article really makes the argument that what differentiates is not sort of this idea of talent, but that there's these habits that these sort of practices that you drill into habit. I'll give you the bad news, right? You're hearing about this now. I saw some of you take pictures of this slide. So the bad news is they do deal with the situation where people have one inspirational moment, and that never works. So it's not about one inspirational moment, right? Because that you might like have good practices for the next week, but you're not going to keep them up. So to continue in this, you really have to, in some ways, take joy in the process and take joy in these little mundane steps.
Do you do that?
Do I take joy in the process? Can you tell? Yeah. Yes, I take some joy in the process. And then they also argue that it's key to, this is sort of what I emphasized earlier, right? Every project, you don't know going in if it's going to be easy or hard. So treat every project the same way. This idea of sort of maintaining that is key. Okay, I'm gonna spend fewer than 4 minutes on this digression. This is a fun project that we just wrapped up in the lab that came out of a conversation I was having at a conference over lunch. Anyone want to guess what breed of dog Rocco is? This is from Doggy DNA, the Reddit. There's a whole Reddit community of people who post their dog DNA tests. So from one test company, Rocco is a bulldog, a pit bull, another name for a pit bull, and a Neapolitan Mastiff. From a different company, Rocco is a Rottweiler, a pit bull, a beagle, a chow, a Labrador, a Russell-type terrier, and a super mutt. These are pretty different, right? So there's a lot of different companies. You can choose. These are 6. And you could say, well, that's really weird, though. When I go to those websites, I have to put a photo of my dog. Why do I have to put a photo of my dog in? They're just testing with DNA, right? So we hatched a plan to try to figure out if the photo actually influences the predictions. So this is a project led by Haley Rando, who was a postdoc in the lab, who's just starting her lab at Smith 18 days ago. And what she did was really simple. So this was our pilot. You can see we like to do these little pilot experiments. So the dog here on the left is Bolby. Bolby is an AKC registered golden retriever. The dog on the right is Robin. Robin is a village dog. What we did is we just took the DNA from Bolby and submitted it with the picture of Robin. So we did this for 6 different test companies: Embark, Wisdom, Oravet, Darwin's Ark, DNA My Dog, and one called ViaGuard. And we got some curious results. So many of the companies are convinced that this DNA from the golden retriever paired with a picture from a village dog is in fact a golden retriever. However, not all companies are so convinced. And so then we designed a larger study. I'll skip this, but we did a bunch of dogs. These are all the good puppies involved. So these are the control dogs. For the control dogs, we ended up submitting DNA and a picture from the same dog. For our treatment dogs, we swapped, and we tried to swap over the phylogenetic tree of dog breeds because we didn't want, we wanted to get dogs from different clades. And then we were like, well, how do you get a photo-based control, right? Because we could just do this with Mechanical Turk and ask people to identify dog breeds. Or it turns out there's just a readily trained neural network that'll predict dog breed for you. As you can see, Bolby is mostly golden retriever, but also part tennis ball. It's close enough. And so then we've got, how do we plot the results? So we plotted them at these ternary plots where you've got DNA only, photo only, something that's neither DNA nor the photo. This is where part DNA, part other, part photo, part DNA falls. And then we can just do this for each of the companies. So this is the neural network that uses only photos. So you can see that it tends to prefer the photos. And then if the DNA for the control dogs, the DNA and the photo are the same, we use the DNA as the axis that's shown. So this predicts by photo. This one is very curious because it appears, sorry, our legal folks at CU have helped me structure what I can say. So these results could be viewed as consistent with a neural network that predicts using photos. The rest do appear to use DNA, at least in part. And I'll just say, if you think of this as like random esoteric work that doesn't actually have any impact in the world, these tests can have financial consequences. So our city council had passed a certain breed ban that had been on the books for 30 years. They repealed that breed ban, and then the mayor actually, this is years ago, but the mayor at the time ended up vetoing the repeal of the breed ban. And so just to give you an idea, according to at least one of the testing companies, Bolby is apparently part pit bull, and so would be restricted under some of these breed bans. And this here is a wolf hybrid. So if you're interested in this, there's more. This QR code will take you to a little bit of a story about it, and this preprint is now up on bioRxiv. With that, I would be happy to take whatever questions you have. And yeah, hopefully some will be about the dogs.